Can you guys hear me? Okay, perfect. Well, thank you so much for having me. Certainly, I think it should be a fun day and packed with new friends and a lot of old friends that I haven't seen in a while. All right, so I'm going to be telling you about some of our work on particular twist supervised learning. Our goal is not necessarily to engineer the best artificial vision system, but really to figure out strategies that would help us build better models of the visual system. Those two goals might or might not overlap necessarily. Before I get into the weeds, I want to give credits where credit is due. Much of the work that I'm going to be telling you about is the work of Drew Lindsley, who is here at the back, who is an assistant professor of research at Brown, and Paizhen Zhu, who is also there, who was an RA all this year and is going to be joining us for his PhD next year. All right. So I like to start my talks with a little demo of what I think is my ultimate favorite experimental paradigm to study primate vision. Many of you probably have seen this demo before. If you did, I apologize in advance. I'll go very quickly. But the demo is about rapid visual categorization. Actually, you can ask who has seen this. Nobody has experienced a rapid visual categorization experiment? Perfect. All right, so you're going to see images flash very briefly. Jan did, yes. So you're going to see images flashed very briefly. The goal there is really to push your visual system to its temporal limits. You'll see images. You'll see, hopefully, things go well. You will see maybe a dominant object, what we routinely call the gist of the scene. But you shouldn't have enough time to see every possible details of these images. So I'm going to start here. So I should point out that this is slowed down to about 100 milliseconds. In the lab, we would shorten that time to a few milliseconds. So why do we care about those kinds of experimental paradigm? You know, studying vision is complex. There's a lot of things going on in vision. And the assumption is that when we push our visual system to its temporal limits, we're going to learn something about the building block of vision, something akin to the very base core representations that your visual system builds before more complex visual routines takes place. For instance, eye movements, shifts of attentions, and things of that sort. So we tend to think as vision scientists as the essence of what vision is. People can solve those kinds of tasks fairly well. You'll make mistakes if you're subjects in the lab, but you'll do, depending on the data set, roughly around 80–90% accuracy. Humans are not the only one capable of solving those tasks. Neuroscientists have been able to train monkeys. So here you see an actual experiment. This is not slowed down for the demo. You see a monkey holding a button, releasing the button and touching the screen whenever it sees an animal holding the button otherwise. The image here goes very fast. In addition, there is some random noise that's being flashed after the stimulus to really kind of push the visual system to its temporal limits. So what do we know about our ability to solve these types of rapid visual categorization tasks? Well, we know that our brain leverages a set of visual areas, routinely called the ventral stream of the visual cortex. You might have heard about area, primary visual area V1, V2, V4. Our visual representations culminate at the level of IT in the temporal cortex. We know that throughout this ventral stream, the visual system builds a hierarchical visual representations, starting with relatively simple visual representations of edges, gratings, things of that sort, and a gradual buildup of both the complexity and invariance of the visual representations. Those visual representations culminate at the level of IT. I'll tell you a little bit more about IT in a few slides. But the assumptions there is that, or the main facts about IT is that one can find in IT neurons responsive for parts of objects or even for ecologically important classes of objects like faces and birds and cars. If you're bird and car experts, you'll find neurons in that part of the brain selectively tuned for those classes of objects that are important. So much of what we know about the anatomy and physiology of this set of areas date back from somewhere around the 60s and 70s. An assumption that I think is now fairly widely validated is that although your visual system, although all these areas of the ventral stream are almost always reciprocally connected with both bottom-up feed-forward and top-down feedback connections, the assumption is that when we push a visual system to its temporal limit through these rapid visual representations, the visual system reaches its fastest possible speed by limiting processing to a single feed-forward sweep through your visual system. So the assumption is that when we push a visual system on this rapid categorization task, there is limited contribution from cortical feedback. In terms of models, I thought I would give you a couple of points here, given that some of the pioneers are here in the room. There's, of course, a long history of models. Fukushima first developed the neocognitron in the 80s based on core neuroscience facts and knowledge. And then I think, one point that I'm going to try to make here is that somewhere around the 90s, the development of computational models of vision or object recognition kind of split along two communities. On the one hand, the computer science community with people like Jan and others really pushed the engineering forward towards building more robust, efficient, performant neural networks. This was the work that led to the modern day convolutional neural networks. But then there's also a whole kind of neuroscience community that worked, in near complete independence, the work that was done in computer science, where models, and error was certainly part of this community, with the development of hierarchical models, both for the ventral stream shape processing, as well as the dorsal stream for motion processing. And so the focus of this neuroscience work really was less about engineering and performance, and rather working with relatively toy problems and stimuli to build in those architectures explicit constraints from neuroscience, from the anatomy and the physiology. And with a goal to summarize large body of data and try to make predictions for experiments. And to some extent, I would say both communities have been quite successful by a number of metrics. But certainly, one great achievement, I think, of the neuroscience models, the so-called hierarchical feed-forward models, is that at the time they were able to integrate and summarize, as I said earlier, a large body of experimental data. And starting from experimental neurophysiology constraints, they were able to show that the pattern of correct and incorrect responses, human behavior on this rapid visual categorization task, was consistent with the behavior of those models. In other words, we were able back then to close the gap from monkey electrophysiology all the way to human behavior during this rapid classification tasks. All the more fascinating since all these models contain essentially 4 key operations that repeat in a cascade. You're all familiar with those operations. This was, the good old days where our models contain convolutions, rectifications, divisive normalization, and pooling, and that was it. Just repeated in a few cascades, and then those models were able to recapitulate a large body of data. So meanwhile, as I said, CNNs, were pushed towards better engineering, and the rest is history. We know that CNNs led to at least the first, I guess we would call, deep learning revolution. And certainly the progress in the field has been tremendous. I'd like to show a little summary of what has been happening over the past 15 years. This comes from Papers with Code, starting in 2012. So this is accuracy of kind of the leading models years after years. Top 1 accuracy on ImageNet as a function of dates. You have your favorite AlexNet here, 2012, roughly around a little bit over 60%, going through different phases of development, leading to modern day deep neural networks that are well above 90%. So, I don't know, actually, I should ask Jan if he would have predicted that, 25 years ago, at least when I was a grad student. Certainly, I wouldn't have expected to see that in my lifetime. So this is a tremendous achievement. And we are in regimes where obviously, these models are starting to compete with human observers in not even speeded presentation when people have all the time in the world to answer. So setting a level of accuracy or comparisons to humans on ImageNet is quite challenging. There are a number of reasons for that. One of them is, and I would say the primary one, is just experimental. It's tedious experimentally to test human subjects on 1,000 alternative force choice. So you need to design clever experiments for that. It takes a lot of time. And the other issue is that, as I'm sure all of you are familiar here in the audience, the labels on ImageNet are pretty noisy. So they were limited to one single label per image. And we know that, on many images, there are multiple objects. Which means that models could be penalized for answering correctly. There's a work that we're particularly fond of in my lab, which is work done in a group at Berkeley a few years ago, where they actually went on and tried to correct and address both of these challenges. So they were able to run experiments for, I think, somewhere around a year to assess human level of accuracy on ImageNet. And they also took a subset of the validation sets and relabeled it with multiple labels to get a cleaner kind of baseline. So there's an extension of ImageNet validation set to incorporate multi-label, for which a human level of accuracy is available. It's hard to compare what's derived from these multi-label versions to the top one accuracy, but what I did here just for illustration, so to be taken with a grain of salt, is that in their paper, they reported that the fixed res next is roughly at human level on the multi-label version. So the red line here is my best guess at where human level would be for this top one accuracy. So, what's pretty obvious is, starting around 2018, potentially all of those models from 2018 are already above human level of accuracy, again, on unspeeded tasks. How representative are those models to, everyday models that we use? We wanted to ask the question, and so Drew Lindsley, who is here in the back, took a libraries of models known as TEAM, which incorporates much of the modern day kind of convolutional neural networks, transformers, a lot of different pre-training. They're all pre-trained and available in PyTorch, so that give us a good sense of where models are today. So Drew took those models, ran them on the multi-label version of ImageNet, and estimated how many of them fall within the human confidence interval. And we found that about 30% of those models fall within human confidence interval. A whooping 4% actually falls outside of the human confidence interval, suggesting that they are already at superhuman level. So this is incredible. This is as I said earlier, a major feat. And if you, like myself, go to the Vision Science Society meeting, which is the main meeting where people studying biological vision meet, you'll casually hear some of my colleagues making statements such as, deep neural networks are currently the best models of primate vision we have, which at a surface level seems like a reasonable thing to assume, but as most of you would expect, just because two vision systems operate at the same level of accuracy does not mean that they leverage the same visual strategies. And in fact, a lot of the remaining of my talk will be about highlighting some of the key differences between modern day deep neural networks, large-scale neural networks, and human vision. But I would say, initially there was a lot of excitement for this move away from the kind of more bottom-up approach to modeling biological vision towards leveraging engineering. Came from early work by Jim DiCarlo, Dan Yemins, and Jim DiCarlo. This is just a figure that I reprinted from one of their review, where they show that indeed, where they show that indeed, so this is a standard. I should maybe take a pause here to make sure that everyone is on board. There's a whole, or the main way in neuroscience whereby people evaluate the goodness of fit or the similarity between representations learned from various kinds of model and neural data is by taking a model, considering individual layers, and then fitting a linear function from the layers unit responses to actual monkey neural responses, who are presented obviously with the same set of images. So these are recordings from the IT cortex, which I alluded to earlier. You see examples of neural responses here. You can quantify the goodness of fit between individual models and neural data. And then in general, when we report the goodness of fit of a given model, this is based on a search through all possible layers and then reporting the best goodness of fit across all layers. And so the initial message that came from computer vision was one where, just letting go of some of the early neuroscience constraints. The HMAX is a model that I was involved in the development back in the years 2000. V2-like is a model that actually Eero, Simon Shetty developed in his group. What you see here, each dot is a model, so categorization performance is on the x-axis. The goodness of fit with IT is here on the y-axis. you see that the message from Yamintz and DiCarlo is that as soon as we let go of some of those neuroscience constraints, we optimize models for performance, we get massive gains, not just in terms of categorization performance, but also in terms of alignment with neural data. So this step or this led the way, paved the way for, I would say, much of modern day visual neuroscience, where the goal here could be to just optimize for accuracy, and then we'll get better models of vision. This led to the work, follow-up work in Jim DiCarlo's lab on brain score. This is just a slightly updated version of that previous figure, where again, ImageNet performance is here on the x-axis. Brain score, which is the measure of goodness of fit between models and IT data. You see that, at least circa 2018, the trend was pretty clear. Better models, more performant models led to better alignment with the brain. So this study was published in 2018. Of course, the field has moved on since, and so we wanted to explore and try to see where the trend stands today. So Drew went back to brain score. So I should point out that brain score is a challenge that was set by the DiCarlo lab. This is a big website where people upload their models. The models are all evaluated on ImageNet, and you have a whole kind of pipeline to evaluate the goodness of fit of those models with against neural data. So this is just a snapshot of one of those data sets, which is an IT data set. And so if we look at the trends, I would say the trend have been changing. So here's a reanalysis from Drew, where every square here corresponds to a different architecture. From brain score. Again, ImageNet top one accuracy is on the x-axis, neural alignment on the y-axis. And so if we think chronologically, models have gotten better and better. So older models are somewhere on the left here in terms of accuracy, newer models on the right. You see that things were going well for us as models were getting better up until the point where they reach 70% accuracy. We were making progress in getting better models of neural data, but since this point here, the trend has been reversing. Yep.
Can you say a few words how you actually measure the neural alignment?
The neural alignment, so I can expand in the Q&A or afterwards if you're interested. But so you have a linear fitting procedure between the layers of the models and individual neural data. And then you just estimate the goodness of fit, and that gets normalized according to what we call the total explainable variance. We have noisy experimental data, and so you can normalize for the noise and give you, based on how reliable the neural responses are, we can estimate a ceiling on what we can expect from the goodness of fit. So the scores are this way normalized, so that a score of 100% means all the explainable variance in the experimental data is accounted for by the models. Is that good enough for now? Or okay. I'm happy to follow up. All right. So I'll speed up a little bit here. Just another data set. This is a data set collected by collaborators of ours at Harvard. This is the lab of Marge Livingstone. Slightly small differences in terms of qualitative differences between those data sets, but this is still IT Cortex. These are models run by Drew, again, based on our team library. And again, the trends here are very similar. You see an improvement, and then a change point, and then the newer, more performant models are systematically becoming worse model of neural data. If we not only are they no longer predicting or fitting better neural data, but when we look at the actual importance of individual factors, so because these are team models, Drew was able to get all kinds of metadata associated with those models, number of parameters, the resolution of the inputs, number of layers, their accuracy on ImageNet, the scale of the data used to train them, whether this was small scale, large scale, whether we use language. And so although there was few factors predicting accuracy during the initial pre-change points, at least accuracy on ImageNet and the size of the training data set tended to be a factor that was accounted for their the goodness of fit of the model. We see that post-change point in red, all the factors are gone. So my argument, to be provocative, is that we're essentially not learning we were learning very little with this exercise of fitting models to experimental data, but we're not learning anything anymore in the sense that there is no factor predicting which model will fit well and which model will not fit as well. Right, one more example, and then I'll switch to something else. This is a data set now of human psychophysics data, so different, I showed you neural data. This is a data set we collected a few years ago. I'll tell you more about this ClickMe, but this is our best attempt at characterizing human visual representations. We have a game called ClickMe, which allows people to select part of an image that they deem important for visual recognition. Again, I'll tell you a little bit more later. But that gives us, in a sense, or that allows us to derive an importance maps from human observers that we can then compare to importance maps derived, with your favorite saliency method from deep neural networks. In the same way, we can plot here for our team models, ImageNet multi-label accuracy versus human alignment, where here we're simply measuring the correlation between the model importance maps and the human importance maps. And we get the same trend again. We find that early models, with early models, there was a clear trend. Now the trend is reversing with more and more recent models getting worse and worse models of human vision. Just want to bring up your attention to this part of the curve here, where you find that the top performing modern models have 0 or negative correlations with humans, meaning that those models and humans will look at opposite part of the image, almost quite literally. And again, same story with the factors. Pre-change, we had a few factors driving these trends. All the factors are gone, so there is nothing else we are learning from more recent models. Okay, now, in terms of what's going on here, one could formulate a number of hypotheses. And I think, just to simplify, there could be two factors happening, two changes happening with modern day deep neural network. One could be that there is an issue in the way we train them, right? That simply they are not learning primate-like visual representations, either defined neurally or behaviorally. Or it could be architectural, right? After all, I made the case, I showed you a wiring diagram of the visual system. There's somewhere around half a dozen processing stages in our visual system. Modern deep neural networks, are orders of magnitude deeper. Perhaps those architectural constraints end up mattering in the end. And that's what's explaining the drawback or the change in the trend. Certainly, I think architectural design is important. Matching and constraints from biology is important, but I don't think this is what's going on here. And the evidence for that comes from work we did a few years ago, where we showed that it's possible to force alignment between modern deep neural network and human feature data. So this is a figure that I took from a paper we published a few years ago, where again, each dot here corresponds to a different model. The set of models is smaller than what we have today, but this is the ImageNet accuracy on the x-axis. This measure of feature alignment with humans on the y-axis. And so what we did here is very simple. We said, well, can we force the alignment between models and humans by extending the loss functions that we use during training? Not only are we training the models by through cross entropy, but also we are adding a penalty term that essentially forces the model to pay attention to image regions that are important for humans. It's relatively straightforward to do this. Importance maps for models is based on the gradients. We can get ground truth data from humans. This is another importance map. And so the gradient is differentiable. So we can take a gradient of a gradient and optimize during training and force the model not just learn to categorize, but to align with human experimental data. So the approach works. It's not that surprising. It just shows that we know how to do optimization. But here you see starting a few representative models that we use as starting point. And you see that when we do this procedure, again, this is obviously separate training, validation, and test. Something like a VGG gets a big boost in terms of alignment. This is just to show you, a few sample images. This is what a VIT would select. This is human data. And after we harmonize, so we call the procedure harmonization. After we harmonize, we find that the models are able to learn a very, or much better aligned with humans. As a side note, the interesting piece here is that I expected the harmonization to come at the cost of accuracy, which in most cases doesn't seem to happen. So there's not a very significant compromise between being human-like and being accurate on ImageNet classification. All right, so what I've told you so far, I've made the point that modern deep nets are becoming increasingly worse models of primate vision. This is bad news for the state of neuroscience. So we need to come up with alternatives. I've shown you that the alignment between models and primates can be improved if we force this alignment with actual data during training. And that seems to be, at least as far as we've been able to test this, we've been successful at harmonization across a number of architectures, suggesting that at least for the prime of alignment, the architecture itself is less important than the way the models are trained. So our key assumption here, and it's probably not going to surprise anyone here, is that we need to rethink the way we train our models. That certainly ImageNet and more generally single image classification is probably not the way to go. So, yep.
So the deep learning models are comparing, are they trained in a supervised way or just random?
So the ones we harmonized here were pre-trained on ImageNet, and then they get so the comparison is with models pre-trained with ImageNet. Here we are forcing them to align. I'll show you some of our more recent work towards the end, where we start from, where the goal will be to use self-supervised learning to yield those models to better align with humans.
But the alignment figures you showed previously, are they?
So there's everything, right? Yeah. So I'm going pretty briefly, quickly. This is the legend. So there is anything from a CNN trained on ImageNet 1K. This is team. So there's a wide range that also includes things that have been pre-trained with self-supervision. There are some larger data sets. I think some of the models were pre-trained on Lion. There's really a lot of things. Essentially, I think all that part here that sucks is really, all the latest and greatest.
Thanks.
Yeah. All right. So the assumption we're working with here is that we need to rethink the way we train our models. And so we're going to be trying to emulate something that looks more like the kinds of visual experience and learning that human babies and primate babies experience. So the work that I'm about to describe will be about trying to figure out better data diets, objective functions, and more generally learning principles that would yield those models to better align with primate vision without having to bake this in as an explicit constraint. All right, so I'm going to go quick because I'm going to run out of time. But all right, so talking about baby-like data diets, there's a lot of available data sets already. One of my favorites was when I was back when I was a grad student. People were trying to get ecologically valid data from cats. So there was a cat cam, which essentially was a bunch of grad students hooking up a webcam to the front head of a cat running in the woods. But because the camera had to be connected to a laptop, there was an experimenter, running around behind the cat with a backpack, holding a laptop, etc. So we're not going to work with a cat cam. There are baby cam alternatives. Sorry. So there's a number of alternatives, including some data we used to collect a few years ago with my colleague Dima Amso. So what you see here is a baby with a portable eye tracker. So the target here corresponds to where the baby is looking at. Here the baby was put with a bag of new toys. And so what you see here is the baby experiencing, this new objects for the first time in their life. And so what you see here, just to illustrate some of the points I'm going to be making, you see the baby kind of, grabbing, manipulating objects, and essentially experiencing a much richer visual experience than what our model presented with IID samples would experience. So there's a lot of work, obviously, focusing on training deep networks on those baby cams. We decided not to work with baby cams for a number of reasons, but most importantly, because those data sets tend to be small, relatively biased in the choice of objects, and they tend to also be not very well controlled. You get a little bit of everything in those videos. Instead, we wanted to have more control over our data diet. And so we went for a publicly available data set called Code3D, which is essentially people collecting short videos around objects with their cell phones. And one benefit of this Code3D is that it comes with a bunch of NERF models that are already pre-trained, which means that we can use those models to regenerate novel sequences for which we have very good control over the range of transformations that is present in the video. So we can generate translation, rescaling, 3D rotations, and things of that sort, and at the very least understand the correspondence between what's being fed to our models in terms of transformation sequences and what's being learned as a byproduct. All right, so finding the right learning objective, again, could take me an hour to go through all the previous work. There's a long line of work focusing on learning better visual representation. For people in the audience who are old enough to know this classic work from the 90s, there was a lot of work on slow feature analysis. This was and variants, starting with Foldiak trace learning rule. You might have heard about the slow feature analysis from Viscott and Sejonowski. The idea was to leverage smooth transformation sequences, temporal continuity across frames to learn better, smoother, more invariant visual representations. There is a newer versions, updates on those. You might have heard about predictive coding. There's very good work from Gabriel Kraman and Dave Cox. Aero has proposed an alternative, which is and closely related, I'm assuming, called perceptual straightening from video data, and many other kind of self-supervised algorithms from neuroscience that I'm not going to tell you much about. Our focus will be on more trying to adapt kind of current and common methods from computer vision, things like mask autoencoders and kind of teacher-student methods, which I'm sure we'll hear a lot about today for the rest of the day. Okay, so our goal is to go beyond the idiosyncratic parameter choice of methods that have been published. We would like to discover kind of general principles that would help us figure out how to better align models to humans. So the space is large, and so the search will be daunting, but we came up with an initial taxonomy or organization of the space to help us kind of at least organize our thoughts. So the general approach will be using versions of mask autoencoders, so we'll hide part of an image to try to reconstruct what's happening underneath, and we're going to be doing that over space and time. And so there are two dimensions here that matter. The first one is the mask coverage, and the other one is the temporal dispersion of masks. So maybe to get you organized here on this plot, I'll start with maybe the simpler example, which would be the autoencoder. We think of the autoencoder as just reconstructing a single unmasked frame. So it falls here on this 2D plot at 0% mask and just kind of a present frame to be reconstructed. But if you want to think about something like a mask autoencoder, if you input a sequence of multiple frames, you can scatter the mask and the parts of the sequence to be reconstructed. And so you fall kind of somewhere around the center here in terms of this y-axis, because the patches cover the entire sequence. And the mask coverage is also in the middle because it's a fixed kind of percentage. We can also represent an autoregressive model here in this diagram. So if you have an autoregressive model, you're going to be focusing. So this is what we commonly refer to as predictive coding, I guess, in neuroscience. You have a sequence of frames and you want to predict the next frame. And so this falls at, it's a full frame to be reconstructed. So it's here at 100%, and we're trying to predict the future. And so when you think about it, most framing of self-supervised learning will fall as one data point on this plot. And then obviously the idea is to pass this organized input into an encoder and then to reconstruct the image behind. Another third dimension is how you reconstruct, whether you reconstruct in the pixel space or in the latent space. And here again, we're going to try to contrast different versions of those algorithms. All right, so this is in terms of data diet and objective function. We need a way to characterize the visual strategies learned by models and how they compare to human visual representation. So I already mentioned very briefly, I have 10 minutes left. I mentioned a little bit about ClickMe. I guess maybe I'll show you a demo in the Q&A if I have time, but this is the old interface of our ClickMe. If you were a player, you would come to our website, you get an image with a class label, and then you are told that you're a teacher and have to teach or allow a student to recognize the image. So the students start from a completely blank screen. And so as a teacher, you have to decide what part of the image to reveal gradually over time. The students start from a blank screen, and then the image gets revealed gradually. The players make points based on how quickly the student recognizes the class label. And so the assumption here is that for players to do well, they have to introspect and ask themselves, what are the most informative pixels in this image, and then decide what to reveal for the student to recognize quickly. We can talk about ClickMe in the Q&A. We've done a lot of controls. The maps we derive are reproducible across subjects. We've also extensively validated the approach using psychophysics methods. So we are pretty confident that the maps we end up with are relatively representative of features that are important for humans. The initial data set had we collected about 500,000 ClickMe maps. This was back in 2019. We were able to get 200,000 maps averaging out across multiple subjects on part of the validation of ImageNet. And then we are in the process of running an updated version where we're trying to collect all the images for all the click maps for the entire ImageNet. And if you want to play, here's a QR code, and I can give you a demo at the end. So these are the kind of maps we get from this ClickMe. This is for ImageNet. So, as I said, there's quite a bit of consistency across subjects. For animated objects like animals, people will typically select facial components. For things like vehicles, they'll tend to select wheels, the front wheel of trucks and things of that sort. We also run part of the Code3D on this game, and these are the kind of maps we get from Code3D. Again, there is no animated objects on Code3D, so it's all inanimate, but certainly for vehicles and things of that sort, we find kind of good qualitative agreement with what we expect, given what we've learned from ImageNet. Okay, so back to our team models. So here we have each dot correspond to a different model. We have vision transformers in red, CNN in orange, and then hybrids between the two in gray. Classification accuracy on Code3D on the x-axis, and then alignments, again, deriving the importance maps from all the models, and then comparing to human data on the same images. Correlation score, normalize, you get a score between -1 and 1. So the models fall somewhere on this plot. Bulk of the model is around 85% or so, and then we get kind of medium range type of alignments. So what we're going to try to do next is to try to see, so these are all models pre-trained on ImageNet. The question we're going to be asking is, can we discover the right objective function that would allow us or help us steer those models towards more human-like visual representations? I should point out that we are still actively working on this. Paisan was still sending, and Drew were sending results last night. This is just one single model for now. We are running a lot more models and a lot more loss functions, but so to be taken with a grain of salt. All right, so we started with a VIT small. And so if you just do linear probe on a pre-trained VIT small, you get here, okay? So the next logical thing to do is to try to fine-tune the model on Code3D. And so if you do this, you get here, the procedure works, you get an improvement in accuracy, but there's no improvement in terms of alignment with humans, right? We're roughly at the same level. Then you can try to do MAE, right? So mask autoencoders on a single frame. There's no temporal information, no transformation sequence being leveraged. That doesn't seem to be helping us very much. We are doing worse in terms of classification accuracy, and we're doing about as bad in terms of human alignments. This is where a video MAE gets you. So video MAE would be the next best thing you can do after an MAE, which is considering a small 3D temporal volume and then trying to reconstruct what's happening in this volume. And so here you're actually doing worse in terms of learned representation for Code3D classification, but there's a bit of an improvement with human alignment. So the features that are learned, or the direction in feature space towards which this model was steered tend to line up better with human, or align better with humans. And then we have two kind of interesting models. We don't have ton more results to show, but I would say it's encouraging. Those are the autoregressive models. So remember, I described those as the predictive coding style, where you are given a sequence of frames and you have to predict the next frame. And we have two versions here, one where we are trying to do this in the pixel space, and then another one where we do in the latent space. Again, one single architecture, so don't take those comparison of those numbers too seriously. So far on the VIT small, we find that the pixel reconstruction ends up helping align better with humans at a little bit of a cost in terms of accuracy, while the latent version, so the student-teacher version, yields a smaller alignment or improvement in alignment with humans, but a slight improvement in terms of classification accuracy. Yeah.
Sorry, one question. So when you said in our probe, what is the pre-training here?
So these are all pre-trained from ImageNet. So we are doing something funny for a self-supervised learning audience. Here we're not using self-supervised learning for pre-training. We're just asking, we're starting from already decent models. Can we improve the alignment? Can we steer those models towards better alignment with humans? We can talk about why we do this later if you're interested, but I'm sure you can guess. Okay. All right. Okay, so now good news is that, so this is again one model. We're running more experiments at greater scale, but those results, those trends seem to hold onto ImageNet, meaning that whatever we do based on this relatively small and limited Code3D yields to similar trend on ImageNet. So again, this would be what the fine-tuned models do on ImageNet, and then you see that both the autoregressive pixels and latent. So the features learned from Code3D generalized to ImageNet in terms of improving the alignment from humans. So I think this is pretty encouraging. I'm going to skip that. We can look at those if you want later, but this is just to give you a sense of, these are human click-me maps, and these are what these autoregressive models do versus alternatives. I mean, in general, we find that anything from a linear probe to a fine-tuning to a video MAE, or at least to a fine-tuning, seems to be selecting almost as much of the background than it does select the foreground. As soon as we move into the video domain with video ME, autoregressive models, we tend to select more foreground part of the image. I'll skip that in the interest of time. Okay, so just to conclude the second part, our goal here is really to try to discover the right data diet and learning objective. I've shown you still very preliminary results, but I think they are encouraging. We've seen that learning from object transformation sequences to predict future latent, potentially sensory inputs, something akin to predictive coding in neuroscience, yields visual representation that seem to better align with human perception. This is still very early. I wish I could tell you qualitatively what's different about the learned visual representation, and especially in terms that would speak to a vision scientist. There are things like, we know the human or the primate visual system is sensitive to things like non-accidental properties, different kinds of junctions that gives our visual system different kinds of information about the properties of 3D shape information or 3D surfaces. We don't know yet, but we're working on this. All we can say right now is that there is a quantitative improvement. And then, just to conclude and to do a little bit of advertising for Drew's talk, lightning talk this afternoon. I think if we really want to make progress in our field on the biological vision side of things, I think our 2D benchmarks or 2D image-based benchmarks are completely saturated. And so I think if we really want to make progress, we need to start rethinking our challenges towards more 3D, 3D structure of objects, 3D understanding in scene, to really kind of tease apart, differences between models and humans. And Drew will show you some initial work we've done in that space this afternoon. And then I'll just leave it at that with acknowledgements for all the wonderful people in the lab that have contributed to this work and our founders. Thank you.